I don't know what happened. Computer glitch, I guess, but we're going to say that it's fine now. Cool. So are we started?
Yep, ready to go.
Alrighty, let me just throw my screen sharing on. We're good to go. Okay. All right, y'all. Sorry about the 3-minute delay, I guess. My computer decided to have an update. Got the Windows. But anyways, hello everyone out there who I can see the other panelists, but I can't see all of you. So hopefully y'all are watching intently. I'm Joseph. I'm a graduate student at UC Berkeley Department of Physics. And today I'm going to talk about a topic very near and dear to my heart, related to the molecular highway network, which I've entitled Driving Life, because I want to tell you about how biophysicists understand this crazy highway network. Hopefully by the end of the talk, you'll see what these little dots are and why I like to think of them as just a fancy roadway that's happening at the micron scale instead of at the meter scale. So, to summarize what I'm going to talk about today, because I always think it helps to see what's going on. Oh, I was going to have my laser pointer out. Let me get that ready. There we go. We're going to talk about first, what is biophysics? I think a lot of the talks have been, very nuclear particle focused overall. There's been some math. There's been a lot of good talks, but I want to specifically give this opportunity to talk about what biophysics means to me. I want to talk about why cellular transport is interesting. So why I think it's a very cool topic and why it is necessary to sustain life. Then we'll talk about the components of cellular transport, just a brief overview of like what things are causing things to move in the cell. And then we'll have a quick caveat into how biology is kind of studied in the scientific world. And finally, I'll talk a little bit about my specific research topic that I've been working, as a hard grad student on for the last couple of years, about how the cell directs transport. So, and, these are what I want to go over, but I encourage people to feel free to ask questions as we go along. In the webinar, the Q&A section, I'd love to answer questions as they go along. It's sometimes a little easier than having to hold on and go all the way back. So feel free to get to that Q&A button whenever you want to. So let's start with, what is biophysics? So biophysics often shows us cool movies like this one. These are, mitotic spindles being formed and pulling apart chromosomes into daughter cells. And I think these movies are fascinating. It's cool that we have the technology to both label and then see these sorts of very complex processes occurring. And I think when I first joined, undergrad, when I first enrolled in my undergraduate department at Michigan State University, I just thought biology was this. I thought it was a bunch of random, memorization cycle techniques in order to put together processes. It was all this sort of, have to understand chemical compounds, have to understand which reaction comes after which. And this was, very complex to me, as well as, this sort of, you could also think of it on a broader term of scale where you see things like the human anatomy and you have to memorize a bunch of muscles. And I don't know, to me, that was not very fun. And when I saw like something like physics, where they just kind of ignored these sorts of problems with, all the little minute details of how something can happen. And you just say, okay, this person is going to push a box. What are the energetic constraints? What are the things that you can say? What force? All these sorts of things. were brought up in class, and I thought that was a lot more appealing, to be able to solve something very easily, quantitatively, and be able to reach some sort of direct conclusion from that, rather than all these sorts of complex processes that were shown. And I think that that was the reason that I was drawn to physics originally. So then I joined physics, but from there, my physical intuition drifted me back to biology. And because I think there's a lot of interesting problems to study, but to kind of address that sort of interesting part, I want to kind of harp on how we understand, how different people can understand these sorts of questions. So in this sort of person moving the box example, I like to think biologists would like to ask, how does a human move this box, right? There are many different pathways. You know, you have muscles activated. How do those muscles get activated? What molecular motors are working? What motors are actually moving the cell? What cells are being divided? What cells are being broken, right? To build this muscle, to be able to push this box at some force. I think that is a lot of biology questions. Physics often simplifies the problem. They often ask, well, what fundamental forces and limits exist in moving this box? So the box is, just some box with some sort of properties. It's got a mass, it's got some sort of forces applied to it. And then the human is providing some of those forces, and it's providing work and energy in order to do that sort of action, as well as working against like friction. And those are kind of simplistic ways to broaden our scale. And so I think this is a nice illustration then of when we think about biophysics, we're using these types of simplifying physical principles to, explain and comprehend what biology is trying to tell us. We are trying to apply our, simple intuition, our approximations, our averages, and our simple fundamental limits in order to comprehend maybe some trends in biology, to be able to think about, well, what is biology actually going to tell us, and discover new things about biology through that lens, through more of a, bottom, like from a first principle scale to build up biology. And so you might ask yourself, well, what sorts of questions does this lead to? So one of these questions could be something as simple as, starting at a very small scale with like proteins, which are the basic building blocks of life, and seeing how this protein can get pulled apart, right? What are the energetics that allow this protein to, stay in a closed conformation or get pulled apart? These things happen at very small time scales, like in the milliseconds range. And, these are only tens of nanometers. And this is just a simulation of a ubiquitin molecule getting pulled at its ends. We could also think of something a little bit bigger and a little bit longer in time scale, such as a motor protein. This is a little famous cartoon of kinesin walking on a microtubule. And this is something else we can try and think of, which is happening, at more of the scale of maybe hundreds of nanometers. Each step's about 8 nanometers. And then having its vesicle being pulled. And so this is like another type of question we could try and ask. We could also move all the way to that original image I showed of the mitosis and think about those types of questions. But those things happen at, thousands of seconds. A cell divides, at least some bacteria cells divide every 20 minutes, right? So these sorts of things they're at very long time scales and length scales. So biophysics is trying to take, anywhere in these principles and trying to mesh them together, right? We want to use small bottom-up approaches to also explain our larger scale terms, but also trying to explain our larger scale problems in terms of smaller scale ones, and keep going back and forth. And these are, this all comprises life, right? You can keep building up to meters where you get us humans. So 9 orders of magnitude we're trying to span here. And it's a very interesting open world to try and discover. So if we want to talk a little bit more, so let's move on from just what is biophysics in general to what is the cell, right? So the cell is the basic unit of life. You might remember this from 10th grade biology or something. There's a, basic unit of some, we have 6 criteria for life. Don't ask me to name them all, but the cell is the first thing, the smallest thing that can satisfy all those conditions. And the only way that we are able to satisfy these conditions of life is to constantly, produce and maintain a cellular environment that helps us. So this neuron that I've shown on the left is actually not just a stationary molecule, right? It's constantly working to maintain its life in our bodies, in our brains explicitly. And it uses a bunch of organelles, specialized organelles, and a bunch of other, compounds that it brings in kind of like a factory, right? We like to think of the cell as a factory. It is very chaotic. It is always getting a bunch of materials in. It is processing those materials into proteins that will help maintain its life or help the larger organism at large. It will also, do this according to, DNA that is, encoding all these proteins. It'll process those and export them if they need to. But all of this sort of process of making things requires energy. Energy is our fundamental unit of what we need to constrain. So we need energy is basically the root. And this is probably no surprise to anyone. There is an energy currency of the cell, and this is in the form of chemical bonds. And this is mostly carried by ATP, or adenine triphosphate. This is the energy currency of the cell. So it kind of is something that can easily be broken by a lot of different molecules and use that energy in order to drive these sorts of, work type processes. But the problem is that this battery is not very good, right? It's got about 2 charges. Each time you break off one of these phosphates, you basically get 1 charge gone. And by the time you get to AMP, adenine monophosphate, you have basically no battery left. So this is kind of not the best way to carry energy, because if you could picture me like on my phone, and if I was on my phone and I could only do 2 actions before it died, it would be kind of not a very good piece of technology. So you could also think about, well, why don't we just like keep it next to a charger, right? Let's keep it right next to a source of ATP. And this is done by mitochondria. Mitochondria are the things that recharge ATP all the time, offering this readily available source of energy. And so that's great, because now we can go through, this Krebs cycle, electron transport chain, whatever, but whatever is happening in there will eventually give us our ATP, and that ATP is essential for life. So that's great, but the whole cell needs ATP. And so if it's just waiting, so basically we need to distribute energy. This is kind of a weird concept, but we need to not just distribute, materials, we need to distribute the energy to those materials so that they can, do their job. That's like the goal of life. And so how does it get distributed to where it needs to go is a fundamental question in biology that people are trying to answer. And the first null hypothesis, right? So the first thing you might think about is, okay, well, let's just let it move around in the cell, right? That should be sufficient. If the cell's small, if these things just, at the small molecular level, maybe they can just move by themselves. This process you might know as diffusion, or Brownian motion is the other term. But basically Brownian motion and diffusion are slightly different, but I'll explain them. Diffusion is you go from a more concentrated mass, so you drop in, say, like a very concentrated mass of particles, and over time they spread out to kind of fill the liquid volume that they're in. That is diffusion in itself. This is done via random walks we kind of have called Brownian motion, after Robert Brown, who first, subscribed it in a paper. I'm not saying he's the first person that did it, but subscribed to them. And this is at the, small molecular level. These are pollen grains just sitting in water. And due to random fluctuations, they move around in some sort of random motion, and that slowly, spreads them out, because if you have a very concentrated amount, then it'll over time spread into an equilibrium. So that seems like a reasonable first hypothesis for what is happening. Just to be clear, what diffusion actually is describing, right? Diffusion is based on kinetic transfer. It's based on momentum transfer of particles. So particles have energy, just like this little ball bouncing in this box, which is, not the best animation, but it, is colliding with the box, and that's changing its direction and energy. And diffusion is just this on a massive time scale, or massive particle scale, right? So you can take a particle in a box, and if you look at this particle in a box, you can follow its trajectory, and you'll see that as it interacts with its neighboring particles, it will move around and sample kind of a space around its trajectory. We don't really, and so we could, describe this if we, had all these molecular details, but that's kind of beyond the point. we want to we understand what the processes that is causing this Brownian motion / diffusion. It's a way to transport this particle around. But one thing that might be interesting is, okay, well, this is great. It's transporting our particle, so we're done. But I ask a question, which is, how long would it take for this particle to find its way to the corner? And this might seem like a simple question, right? Like, I don't you might just say, yeah, it'll find its way to the corner eventually. But I'm asking you for that time frame. How long will it take for this particle to get all the way over here? And further, you could ask, well, how about this other particle? What about this little pollen grain sitting around here? How long is it going to take to get to this corner? I want to know those in some sort of quantitative detail, because it's easy enough to say it will eventually, but time is actually an important factor here. So we should figure out, so how are we going to describe this sort of motion? Because unless I give you all the molecular details of every single atom, you can't tell me what the trajectory is, right? That's going to be a hard system to evolve. Well, this is where we get saved by statistics. So we can ask ourselves, well, what if we, instead of describing just one particle trajectory and just like having everything else be random, since we have this randomness, what if we average out over those parameters and just describe our system as a probability? We have a probability of being in any given spot in this system. And if we want to do this, we can actually think about the particle as diffusing as like a Gaussian. It'll actually approximate to a random walk graph becomes a binomial distribution, and in the limit of many particles where the random walk is not equal, you actually get a Gaussian-like distribution of diffusion. And so over time, you'll see that this Gaussian will slowly collapse from being very pointed at a certain spot, and over time it will slowly relax to cover most of the area. Still mean zero, but so the mean is still zero. It will always be around the same spot in terms of the mean probability position. But over time, you'll see that this standard deviation, the variance of the Gaussian, actually spreads out. That is how we can quantify the system. And so we can call this, basically, we can just equate our standard deviation, our variance, to related to the time as well as the distance, the diffusion constant, not the distance, apologies. So the diffusion constant, which is going to equate how much time has passed to how much, mean squared displacement have I done. And so if we want to actually quantify this, there's actually a molecular system that we can look at. And this was described by Einstein, the Einstein-Stokes equation. So it took both Stokes and Einstein, two brilliant scientists, to come up with a very nice and simple relation for how this diffusion constant depends on certain parameters that we're interested in. The diffusion constant in this case is proportional to the energy. So if you have more energy in a system, which in our case is described by temperature, you will have more random motions, which will allow your particle to move farther. That makes sense. But then it is inversely proportional to both the viscosity. So something that is, more viscous, like molasses, is not going to allow particles inside of it to diffuse faster. And also the radius of the particle. It turns out that when your particle has a larger radius, these more random motions of all these particles transferring energy leads to the particle actually being more likely to stay around zero and not as likely to Brownian motion out to a very far distance. So good, we have a relation now. We have a quantitative model that we can now use to ask the question, well, is diffusion a form of transport that will sustain life? So let's think about a diffusion constant of, say, a couple molecules we know. So ATP, I just mentioned it. Well, this is actually ADP, but the ATP maybe has a radius of like 1 nanometer. It's a very small molecule, right? So it can diffuse pretty fast at 200 microns squared per second. If we look at something a little bit bigger, like a mitochondria, which is almost a micron, 1,000 times larger in radius, this can be, 0.2 microns squared per second. So this makes sense because if this is R is 1,000 times larger, then that means our diffusion constant will increase by a factor or decrease by a factor of 1,000. Cool. So using some math here, and I just put in some numbers for T and eta that makes sense for water and room temperature respectively. So just as an idea. And now let's ask ourselves, well, okay, these are numbers, but they don't really tell me how much time it's going to take for me to diffuse a certain distance. So if diffusion can sustain life, then we ask ourselves, okay, let's look at what type of cells we usually have. So we'll start with the bacteria, a little E. coli bacteria, which is actually about the same size as the mitochondria. So it's about 1 micron in radius. So in this case, if we wanted to ask for the ATP time of diffusion, we just take that diffusion constant that I calculated before, and then we know how long we need to diffuse around inside this particle, right? So if I want an ATP to diffuse from, the center of the particle to the exterior, that's the 1 micron or whatever, then we can plug that in for sigma, and we would find that we get 1 micron squared divided by 2d. d was whatever it was, 400 or something, and you'll get about 2.5 milliseconds. 2.5 milliseconds, that's pretty fast. That's like, very snappy. I could get ATP pretty quickly. And yeah, that actually seems reasonable. I wouldn't think I need too much more than that in that case. And yeah, bacteria don't really need any sort of other mechanisms to get energy to the parts of the cell they need to. Okay, let's make the scale a little bigger. What about an animal cell? So an animal cell, we'll say, is about 30 microns in radius. So that's about square root of 1,000 times bigger square root of 1,000 times larger radius. So if that's the case, maybe I'll just have a quick little pause here for an activity at home, which is if you have a square root of 1,000 larger R, then that'll change your diffusion constant by square root of 1,000 divided by square root of 1,000. So what should be the new amount of time that it would take ATP to diffuse? We'll give it a quick 30-second break so I can take a drink of water, and maybe if you're brave enough, you can put it in the Q&A chat, and I will reward you greatly if you can get the answer correctly. All right. No takers. No one wants to venture a guess. That's okay. All right, no problem. I'll give you the first one. Okay, that was a little bit of a tough ask, I guess. So if we think about square root of 1,000 times smaller d, then that means that our time that we would solve for, we would just, once again, oh shoot, I messed up. I'm so sorry, guys. I set you up for failure there. My bad. Not the diffusion constant doesn't change because the radius of our particle doesn't change. Silly me. It's the amount of distance I have to travel. I'm sorry. See, messing up my own math here. But basically, take the square root of 1,000 of sigma. That's what I wanted to say. So that means you're going to have an 1,000 times larger side here, and you have to divide by the same diffusion constant. My bad, y'all. But in this case, then you actually get that the time should be 1,000 times larger. Okay, that makes more sense. Whew, confused myself there early in the morning here. But once you get this time, you'll actually get that then this is 1,000 times larger. So indeed, the ATP diffuses at 2.5 seconds approximately is what its first standard deviation would be. And so you could imagine that that's a little slow, right? That's, if I need to do some sort of process, 2.5 seconds, that's a long time to kind of have to wait between every click of something I have to do, So that's kind of crazy. And mitochondria, which, has a 1,000 times larger diffusion constant, that one actually has an 1,000 times larger diffusion constant. I'm not lying about that one. That one is about 40 minutes. So that's about 2,500 seconds, which is approximately 40 minutes. So at that point, it takes like an hour to get yourself a steady charge. You could probably maybe handle that, but it's starting to get a little less efficient. Okay, now I've set you up with all the things you need to know for a neuron. So a neuron is a 1 millimeter long thing. It's very long, right? At least it's a millimeter. Some neurons are even a meter long. That's crazy, right? A single cell that's almost a meter long. But in this case, so this neuron right here is a millimeter long or whatever. So that's 1,000, another square root of 1,000 times bigger than an animal cell. That's, 1,000 times larger than your bacteria. So how many, seconds would it take for an ATP to diffuse in this case? Don't think. Maybe I could give it like 10 more seconds, see if people are ready for this one now. But understandably, just think about the math. I want to take 1000 microns, put it into our sigma, and then keep our diffusion constants the same. And we'll end up seeing that T increases by a factor of 1000 from the animal cell. So we actually see that ATP now, to go from one, radius part of the cell to the other, would take nearly 2500 seconds, which is about 40 minutes, the same as that mitochondria in the previous example. Factors of 1000 carry over. But this is, almost at the point where, if I could only get a charge every 40 minutes, I mean, am I even doing anything? I'm sitting around for most of the time. This doesn't seem like a very sustainable way to produce life. And mitochondria, if I wanted like a steady charge to get somewhere, just by diffusion alone would take almost 30 days. It would take almost a month to move this, 1 micron sized particle just a millimeter. That's pretty crazy. And this is in water. This is just an estimate from water. Cytoplasm is actually even more viscous. So that's going to decrease it more. And these things have to, and so it's just, this is unsustainable. Diffusion is not the way we can sustain life in the human body. So hopefully you all enjoyed that little order of magnitude exercise using equations. I know someone gave a talk about these sorts of things in REYES, I think a couple weeks ago. I thought it was really interesting, and I was really excited to be able to do this in front of you. So diffusion cannot sustain life. I've hopefully proven that point with the last 20 minutes of my talk. So what is the solution? The solution is we actually have intracellular transport. Intra means, within the cell. And this is a live image, cool biology image taken of actual vesicles getting moved along these highway networks that I was talking about. You can see they're moving both out of the cell body and into the rest of the cell. Oh no, I'm trying to keep it playing. But yeah, and so these sorts of movies are like happening all the time in your cells, where these cargoes are constantly getting moved around. These vesicles are getting moved around in your cell, kind of looking like a little bit of a, network system of highways as you're watching these sorts of particles get moved around. And so this is the way that the cell overcomes a sort of problem. Now, these particles can move very quickly. And if they move very quickly along this particle, they can direct transport within the span of, they can move at like a micron per second. So within 1 second, you cover the distance that, it took a normal, so a millimeter, right? That's 1000 microns. So in 1000 seconds, you can cover the distance with a mitochondria that would usually take, a whole month to do. It's helpful. It makes sense. And this makes sense too. So that's not where I want to go. There we go. And the most valuable thing, I guess, about this intracellular transport network is that it is super important to the sustaining of life as it is. And when it fails, and when other things fail in the intracellular transport network, we get a great myriad of neurological and muscular and currently untreatable diseases, such as Alzheimer's, spastic paraplegia, ALS, Parkinson's, Charcot-Marie-Tooth, Lissencephaly, Huntington's, spinal muscular atrophy, and if not, death. So all these sorts of failures are super valuable to maintaining life as we know it. So once again, maybe a good point, I'll just take another drink of water, pause, because soon we're going to start talking about, well, what is actually the components of this intercellular transport network in just a second. Okay. That's all right. We will still have time for Q&A after all this, so that's for sure. Okay, so hopefully I'm not losing anyone right now, because now we're going into the biology part, I guess. Ooh. Oh no. That's not good. Oh, are we back? Oh, thank goodness. Okay, sorry, my computer was weird for a second. But we're back. Okay, whew. I was scared for a second. Okay, so let's talk about the components of intercellular transport. So this is a little bit biology heavy, and I'm not going to go through all the details, but I think it helps to remember that biology is complicated, but we can start thinking about our concepts in terms of a more, standard classification technique. So what we're going to go over here are the parts of transporting a cargo, which is, the package, the thing you want to get somewhere. So in this process, we have sort of a track called the microtubule, which has microtubule associated proteins that are kind of architecturing, building the track for you. They also have the motors that then actually convert that energy into mechanochemical work to lead to unidirectional transport. And then they are usually adapted to a cargo by some sort of hitch protein that actually connects to the cargo. So these are like these are the things you have to think about when you think of intercellular transport. Is there's just, and this kind of makes sense, right? If I was thinking about this as like a car, this is the road. I have like the workers who built the road and are keeping the road together. And then you have like your actual car, which then has some sort of hitch that's transporting whatever you want to do. But in terms of biology, let's fill in the details with some more, biological systems. There is the microtubule track, which is basically just a large cylinder. So it's kind of cool. It's got multiple lanes, and it's a large cylinder of many proteins that come together. And these proteins come together to they don't just come together usually they will come together on their own, but they're often very dynamic, and they don't want to stay together very much. They don't want to stay glued for super long all the time. So there's structural microtubule associated proteins that come around, and these microtubule associated proteins are things that help stabilize that microtubule. They keep it in a good spot. And then this way, when you have a motor walking on the microtubule, it doesn't just come undone, right? It doesn't just suddenly decay. So these proteins, there are specifically two types of proteins, which I think are fascinating. One type of protein is kinesin, and it moves towards the plus end of the microtubule, which basically just means the outer end of the microtubule, away from the cell center. And then you have this other motor, dynein, which comes in from and goes to the minus end. So you kind of have like a train in this way, because you have, the one facing head, and it can only move in one direction. And then if it wants to switch directions, it has to switch to another train that has to go to the other direction. And so these sorts of transports take both of those systems around. And finally, you have to think about, okay, well, then you also need to have, other than this motor converting making the actual work, you actually need to get a hitch, right? You need to find a sort of mechanism to actually hitch on to the cargo. And so that's demonstrated actually in this complex here. This is dynein with a specific cargo adapter called BIC-DR1, but I don't really care how biologists name things. It's just a cargo adapter. And this cargo adapter will come in, and it will then be able to further talk to the cargo, right? So in this little cartoon, there's actually another cargo adapter called track, which is directly bound to our kinesin motor, and it goes through some sort of other pathways with some other sorts of protein complexes to actually attach to the mitochondria. And this is something that, if you were so inclined to want to read about something, it's something that was published in this paper that I've referenced. So those are the basic essential components of intracellular transport. So great. So we know how I can take a motor and I can start moving it around. But after all these sorts of questions that I've asked, I bring you to a very open question in biology, which is, how does the cell direct the transport, right? This is something that I haven't explained yet. I said, okay, this is how it does it, right? I explained how I can move something from one spot to another, in some sort of detail. Obviously, there's a lot of small in-between parts that I could talk about, but a very open question in biology is very relevant, is how does the cell actually direct transport, right? So, because I guess motors don't have the ability to pull up the destination on their phone, their little GPS, and be able to say, oh yeah, I need to go from here to deliver my mitochondria, a couple, tens of microns away. It doesn't know that. So how does it figure it out? Well, in, biology, chemistry, we're all about trying to understand the signaling pathways. We're all about trying to understand the, there's a bunch, not just signaling pathways, but the things that are close and interacting, right? So these are not signaling pathways, but the things that actually are interacting with the motor at any given point could be solutions to how it's being directed. Because we're assuming that the motor is ubiquitous throughout the cell, and the only thing that changes is the sorts of proteins that it's interacting with at any given point. And so what does it interact with? You could go through the list of what I just talked about. Well, it's always walking on microtubules. It doesn't, if it's off of the microtubule, it's not walking. So these microtubules must be something that it interacts with. That could be a source of direction if the microtubules could somehow direct them. It could also be these microtubule associated proteins, these track architects that I mentioned before, because they're always on the microtubule, they're always interacting. So that could be something there. It could also be the actual cargo adapter, right? If the cargo is attached to a specific cargo adapter, maybe that's the source of, telling the protein how it has to move. So these are all possibilities to answer this question. But of course, there's a bunch of maps, there's a bunch of cargo adapters, there's different modifications to microtubules that are still trying to be categorized. But this open question is, like I said, it's complex. There's a lot of things happening in this question, but it's an unanswered question. So it's very biologically interesting and motivated. So maybe we ask ourselves, well, how can we study a question like this one, right? Because I don't know, I'm, like I said, not a biologist by any means. Last biology class I took in 10th grade was in 10th grade. So and I could try and learn some techniques for biology, but I kind of want to understand it from a more experimental point of view, right? I want to understand how I can actually perform these experiments. So how do we study biology is then the question. And so there's a bunch of techniques, right? I'm not gonna go through like a list of biologically relevant techniques that people use. Instead, I'm going to talk a little bit more about, what the general categories are. So one is that we could actually visualize these proteins in the cell, kind of like that movie I showed before, kind of like the one that I'm showing here, where we can actually just look into a live cell and we can observe, what is happening in the actual cell, where it's actually supposed to be happening, which is great because now you get it closest to the, natural context that it's happening. However, the natural context also is exposed to all those complications that I just mentioned. So you don't have as much control over what is happening in your system. You could do kind of an opposite method, which is, okay, what if we just simulate things in a computer in silico, right? So we could, in silico, go in and try and model our whole neuron and model transport and model all the components of it, which is relevant. It's a simulation, but one, computationally expensive. Two, it's actually almost impossible to model anything this big in the molecular detail that you might want at this point in our career. Maybe soon will be okay. And three, you have to worry about the actual model shortcomings in a computer, right? If you make an approximation and it's wrong, then you're prone to have errors in that case. So these are both techniques that can be used, and they've led to very interesting questions. But the one I'm going to talk about specifically is one that's very popular in biophysics, which is also in vitro, which is in the glass, right? So we can basically reconstitute a system like the in vivo one, but take out all those extra factors. We put it into a way where we can start minimally looking at certain proteins and interactions and stuff to start really separating out what the contribution is from each component, kind of more of a bottom-up approach, right? Just kind of like the computer and all these sorts of questions. And so the specific way that we do this with our sort of system I just mentioned, the microtubule motor cargo adapter map system, is we can actually just fix microtubules. We can make our microtubules, we can fix them on the surface of cover glass, and then we can just label proteins and we flow them in and they just walk on the microtubule with ATP. It's great. So that's all we have to do. We just flow in our proteins of interest, they'll bind to the microtubules, and then we can watch them move. And you get cool movies like this one. Like this is a real movie I've taken, maybe sped up about 5 times or 10 times. But either way, these are like, each one of these little dots is a motor moving on a microtubule, right? Here's your microtubule, and this is just dots moving on it, moving over to the side. And you can see this unidirectional motion in real time, across this distance of 20 microns or so. I think these are cool movies that I get to luckily see every day. So, I'm very happy with this data. And, here's just more actual images of kinesin on microtubules, right? So these are all microtubule images with motors on them. And yeah, I mean, some of them aren't. These are just kind of my approximations, but these are, me visualizing the molecular highway network in my little cell that I've made. But it's, in vitro. Very cool stuff. Okay. But okay, so you've stuck with me this far. I'm surprised. But let's just have a quick little discussion then in the last like 5 minutes I want to spend, or 5 to 10 minutes, talking a little bit more about what research is really kind of hot in this sort of field, right? I've started with biophysics in general. I've tried to motivate intracellular transport. Let me just bring you into a specific example of something that people have done with these sorts of systems. So one thing I was talking about was the microtubule associated proteins are something that the motors are constantly interacting with, right? So is there a chance that these microtubule associated proteins actually lead to a sort of direction in our transport? So in these cases, what I'm showing are movies where I have the same number of motors like in solution, but there are increasing amounts of MAP7. MAP7 is a specific type of map, right? It's just something that is, bound to the microtubule and it's interacting with the microtubule, but it also seems to have some interaction with kinesin. The way we can understand this is not just by looking at the movies where you can see that clearly there are, more particles in like this middle movie, but also you can look at the actual distance that they run. So the distance, this is a chymograph where you have time in the axis here. So that's time going vertically and distance horizontally. And so you can see that the distance that they travel is just the difference between the first and last point. And the time the velocity can also be then understood as the slope, right? The more horizontal your slope, the higher velocity you have. And so what you can see is that each one of these movers actually has a higher distance of running when it's in MAP7 compared to when it's in, no MAP7 condition. But then when you get too much MAP7, it actually starts to inhibit. There's not nearly as many movers, and they actually start to decrease in velocity. And this is kinesin, by the way. I think I did not say that, but this is an example of kinesin moving on a microtubule. So somehow kinesin is actually being helped by MAP7. It's actually starting to direct transport in a specific direction. And we can quantify these sorts of chymographs and movies by thinking about them in terms of, the number of walkers per, micron per second. So a density of walkers. We can think about how long they're running, and we can quantify that data. And we see both of these cases at a certain concentration of MAP7, you actually see there's a maximum amount of motility before then it decreases over some period of time. And further, you can also see this happen with velocity. The velocity just decreases because of some mechanism. So we need to understand this mechanism at some level. But also, before we understand just that mechanism, what about dynein, right? So that was the other motor. Kinesin's supposed to move in one direction, dynein supposed to move in the other. So is there the same effect with dynein? Is this just helping motors move overall? And the answer to this question is it does not affect dynein the same way. In fact, dynein seems to be strictly inhibited. There's no sort of activation / recruitment mechanism and then inhibition. It's just straight inhibited, which this is fascinatingly like simple, right? If there's just MAP7 in your cells, right, and you wanted to force kinesin to move and not dynein, then somehow when you are MAP7 has a strong interaction with helping kinesin, but not dynein. So you're directing your transport towards a certain part of the cell. This is forming an asymmetric process in just the evolution of what's on your microtubule, which is super, super interesting, right? So, and then one thing people might ask is, okay, well, why is kinesin able to be helped out by MAP7? That's basically the question, right? And you can do this a bunch of different ways. People could study. So people studied it and they saw that there's an interaction somewhere off the microtubule, right? And those are all protein specific and all these sorts of things. But you might ask yourself, well, okay, if they interact off the microtubule, but how's the kinesin getting past the map when they're binding at the same spot on the microtubule, right? I can't just walk past something that is in my way. And so you can ask one of these questions using these in vitro techniques by just saying, okay, well, I can actually observe how kinesin would behave when it's near a map, right? Is it just going to dislodge from the microtubule? Is it going to be able to like dislodge but interact off the microtubule and kind of like take these, maybe use this interaction domain to like kind of sidestep one of these maps? Or does it just like carry the map with it? Does it just, hold on to it and carry it around and, use it to help stabilize, kind of like a tether? Well, we can answer these questions by observing the actual stepping behavior of kinesin. We can get sub-nanometer resolution, and we can look as a motor moves towards the plus end of the microtubule, and we can get plots that actually we can fit steps to, right? So this is the steps of kinesin in the plus end direction, right? So it's moving up. Every step is it moving up in the plus end direction, as well as we can look at its sideways motions, right? We can look at like how it would move along the outside of this protofilament, right? And so in this situation, we see that when there's no maps, we see a very straight walking kinesin. It just takes steps very regularly and has no sideways steps. But as soon as you add in map 7, you see an increased number of sideways steps and backward steps, which are irregular. So this seems to suggest that maps are actually causing kinesin to dislodge from the microtubule and then rebind, but they have this interaction that keeps them tethered to, like a tethered diffusion. So diffusion comes back. Hey, there we go. It's not diffusion where it's free floating anymore. It's actually got some sort of constraint because it is interacting with this map. That is a possible hypothesis. But just to wrap this sort of question up, this possible hypothesis is just experimentally shown, but how about, can we model it? Can this mechanism actually explain that sort of biphasic behavior, right? We got to draw a model and be able to predict from this model that we could get that sort of data, and vice versa, right? This offers us a tool that we can use and understand something at a more fundamental level than just one single motor map interaction, right? We could expand this to other sorts of systems. And it turns out that using some sort of model, which I'm not going to go into great detail, but using some sort of model, we can get those sorts of curves back. We can see this sort of increase in the number of walkers and the run length while decreasing the velocity, right? So we can do that. And it's very, maybe I haven't said it in the most detail because I spent a lot of time talking about some background, but these are the questions that biophysicists are trying to answer, right? And you can see the appeal of generalizing something. So I guess the final thing is, I'll conclude. I will, say about, kind of some rudimentary things, biological processes. I think, as I said, I am not someone who is an expert in biology by any means. I took my last biology class in 10th grade. And I never took a biology class since then, I guess, until I took some like biophysics related classes, right? But straight biology, do not know. But I think that there's a lot of benefit into looking at super complicated systems without that super complicated lens, right? Remembering that we can benefit from approaching a bottom-up explanation, right? By thinking about, well, let's not think about all the complicated things happening, but what are the rudimentary fundamental things that are occurring, right? And being able to approximate and reduce problems and think critically offer a lot of value in any field. I don't know, if, maybe this is a little bit overarching, but I think that, one of the things that makes my degree in physics so appealing is that, you spend a lot of time worrying about these exact problems. How do you reduce a problem? How do you approximate something? When can you take certain limits? Those things are very valuable. And that's something that happens too in engineering, in, all these sorts of STEM fields. So very beneficial in that. And finally, just my last plug, I think biophysics is particularly intriguing from physics, chemistry, and biology because it studies new physics. It's got general applications, right? It's something that the world is still pretty interested in. And it incorporates kind of a unique part of experiments and quantitative modeling, right? They're very hand in hand right now. There's a lot of experiments driving what questions are interesting in biology, which is different than some other fields where oftentimes theory is years ahead of the actual experiment that's going to understand it. So yeah, I think all these sorts of things lead to the fact that, I think biophysics is a good thing to look at. And I just gave you a specific example, but if you have any more questions, I'm happy to take them now. So thank you once again. Um, what did you do? Let's see. So, all right, well, it seems like there's not too many questions. Either I've done so well at answering them throughout the talk, or I lost everyone like immediately. So, I mean, you don't have to ask me questions just about this, if you have any questions about, myself, what biophysics is to me, I'm talking about a little bit, but, or like, my journey to grad school, or, more questions in biology, some interesting thing you read on the internet. Got 5 more minutes, Mm. Oh, wow. Okay. Well, now they're going. Okay. Okay. Excellent. Thank you all. Oh, wait, what? Mm, that's interesting. One came in at a weird time. Okay. So yes. So one person asked in the chat. Oh, yes, these are great. Okay, so someone asked in the chat. Oh, shoot, I was gonna type an answer. I'll see if I can do that. They asked, do you have a book recommendation about this topic? I do actually. The book in general is called Molecular Biology of the Cell. Let me see. Oh, shoot, how do I? Okay, here we go. I can answer it here. So it's called Molecular Biology of the Cell. This is a little bit maybe more technical, but it's written by Rob Phillips. So I'll put that in as well, by Rob Phillips. And you can, I think you can get it online somewhere. It's probably got a, it's only, $80 if you really want to buy it in paperback. But this is a very good book. It's actually also co-written by a Berkeley professor, Hernan Garcia, and they have a lot of good type of physical intuition tools for, how you can start to approach biology from a quantitative like physics background, which is very helpful in this sort of case. There are of course other books, but I think this one is one that I have been recommended before by my physics advisor, and I thought it was good. So, just carrying on the chain of command. Hopefully that answers your question. And then, yeah, someone else had 2 questions. So the first one is about maps. What are they exactly? No, that's fair. I kind of skipped over what maps are. So maps are in, can I still, yeah, there we go. Maps are in essence, they're just these proteins. They're actually very interesting because they're intrinsically disordered proteins. So oftentimes you think of proteins as having like a specific structure, and that structure dictates how they function. But there's been a good revolution recently in that people have found that proteins that are just chains of amino acids actually can change a lot of biology when you modify them. And so these maps are structurally basically only have 2 components though. There's a bunch of maps, and they bind to the microtubule. So where's my nice little animation? Man, I went really far back here. Oh. Oh, one more. There it is. So they basically bind to the microtubule. That's one part, and then they just kind of splay out. So their goal is to structure the microtubule. No problem. I know I went over a lot of this stuff fast. It's a lot of material, but like, yeah, basically just think about them as things that bind to microtubules. That's all you need to know. And then the other question is about how can a graduate with purely physics background get into biophysics? That's a great question. The truth is that, I mean, so what did I do? I did physics. So I did physics in undergrad. I did math as well. So I did physics and math. And I also did some computational modeling. And I don't know, I just, at some level, the question becomes more important than how you're approaching it, right? So like, there are plenty of people working on this exact same system from different backgrounds. There are people in biology, people in chemistry, people in a bunch of, my research even proves that, you can approach this problem from many different directions. And so it's less, when you're in research, it's less about your exact, like, where are you coming from? And it's more about what kind of tools are you applying to the problem, right? And so for me, my physics intuition was I wanted to learn a little bit more about life. I had an interesting experience where I worked in an experimental lab that kind of took physics and just did those protein folding experiments. And I just wanted to understand, something more complicated. So yeah, I think overall it's just pursuing your interests. Like biology needs physicists. It's a fact. Like, not because biology like is on its own bad or anything. It's just that physics really offers a lot of advice. So I think that you'll find, if you are interested in this sort of topic, you just reach out to the appropriate people who are doing, I would recommend doing someone who is like a biophysics person more than like, just a straight biologist. That's what I'm doing, someone who has a physics background who's approaching biology questions. But you'll find that there's less of an activation, there's less of a barrier than you might think. At least it's less of a barrier than going from biology to physics, I think. So hopefully that answers your question. Okay, we got one more question I can finish answering before we have time for the next thing. I think I have 1 more minute. So the last question is, how can chemistry relate to help more understand biophysics? It's my favorite field of science. Oh yes, chemistry is a great language to learn. It is a language that I am still trying to learn because so if you have the chance, take more chemistry classes if you're interested in biology. It can only help you out. And yeah, so the chemistry questions that there's a lot of things with structural organic chemistry that we're talking about here. Also, the fact of the matter is that like a lot of my modeling nowadays kind of relies on some sort of chemistry knowledge. And I like to think that chemistry is an application of physics, right? You take physics describes a simple thing, and then chemistry makes it slightly more complicated. But it also has its own methodologies that actually help you understand, most importantly to me, the statistical processes. So these sorts of modeling questions that I'm asking are very chemistry driven. Like these sorts of rates and stuff like that, that's a very chemistry way to think about this problem. And so, these are the questions that are being answered. So yeah, basically to answer your question quickly, it can relate in so many ways, especially with structural stuff. Chemistry is important, and also with how you think about probability and statistical phenomena, where you're thinking about like what's the mean like way that a process is going to go about. Okay. And yeah. And so hopefully that answers your question. And then finally, I'm sorry, just one more follow-up was about computational physics is needed for biophysics. None. Though I love computational physics, and I'm going to be teaching in a couple minutes about, Python for STEM because it's something I'm super passionate about. But I would say definitely know how to do at least some coding, because you can figure out how to do all these like fancy MD simulations or analysis methods as you go along in the field. But I think that it only helps. Like I said, it's one of the tools that I think is invaluable. And there are people in my lab who don't have computational experience, and that's fine. They still do experiments, but they're doing the experiments that I kind of would rather have them do, and I'd rather do a different experiment, if that makes sense. So computational physics, just having some sort of understanding about coding is super valuable. So hopefully you look forward to that. I definitely like the modeling aspect of biology a lot. So cool. Okay. Thank you all for your questions. I was afraid I'd lost everyone, so I appreciate it. And I think we're 1 minute over. So, and last minute things I need to do. Jeff or Beth, is there anything